All right, let's jump into meiosis. So, meiosis is a very specialized version of cell division. It's not one that you're going to see outside of the gonads. So, you're not going to see a skin cell doing meiosis. You're not going to see a muscle cell or a kidney cell or or a lung cell doing meiosis. Those cells are only capable of doing mitosis, which means making an identical copy for growth or repair. So what's the point of meiosis? It's to make gametes, to make sex cells or reproductive cells. These cells are going to be real special because they're going to carry genetic material that is set up in a combination that the universe has literally never seen before. So if you were to take a look at a sperm sample, if we were to genetically sequence every single sperm cell in a sample, they would all be unique. If we looked at every ovum that a woman released from follicles during her entire lifetime, they would all be genetically unique. Yes, you got the DNA from your parents, but you shuffle it in two very important ways in order to make these unique gametes. So it happens over two phases. We call them meiosis 1 and meiosis 2. Meiosis 1 is really the thing that's interesting and complicated. Meiosis 2 it's going to look a lot like mitosis. And so we're going to have daughter cells that are genetically distinct and haploid. So let's jump into meiosis. Let me get a thinner pen here. All right. So we still have PMAT, prophase, metaphase, anaphase, telophase. So what I want to do is look inside of this cell. Now we've already done S phase. We've already copied our chromosomes. You can see that they're kind of doubled up little butterfly or X shapes. But something interesting is happening here that we did not see in prophase in mitosis. In prophase in mitosis, we just condensed all the chromosomes, we got rid of our nuclear membrane, and that was kind of it. Oh, and also the spindle fibers attached, which is happening in late prophase. Here, you can see a little bit of weirdness going on. It looks like the red chromosome. So this would be, the big ones would be called chromosome 1. So we've got a red chromosome 1 and a blue chromosome 1. Remember, these are called homologous chromosomes. So big red and big blue, those are homologous to each other. Medium red and medium blue, they are homologous to each other. And little red and little blue, those are homologous to each other. So you're never, ever, ever, ever going to have big red crossing over or hooking up with little blue because they have different genes on them. And that would be a disaster. Actually, you do see this happening sometimes, but it's wrong and bad, and it causes horrible, horrible mutations and generally cells that are non-viable, that won't survive. But that's an aside. So we've got big red, that's chromosome 1 in red, crossing over with chromosome 1 in blue. What that means is that they are swapping genetic material. And that's okay, that's allowed. They're allowed to do that because they have the same genes on them. For example, maybe on chromosome 1 over here, there is a gene for hair color. I don't know. Maybe you got the red one from your dad and the blue one from your mom. So your mom gave you or contributed this big blue chromosome, and it's got certain genes on it, and they're homologous, meaning that they are the same genes They might be different versions of the genes, but they code for the same thing. It's like we talk about different versions of genes, we call them alleles. We'll talk about that later. But an allele is basically like a recipe. So think about, I don't know, chicken soup. There are many different versions of chicken soup that you could do. You could do just straight up chicken noodle soup. You could do chicken soup with rice. You could do chicken soup with like different kinds of noodles. So there's variations. They're all chicken soup. If you gave them to somebody, they'd be like, yeah, this is chicken soup. It's made out of chicken broth. There's veg in there. There's chicken pieces in there. Boom, chicken soup. But they are not exactly the same. So you could have the gene or the allele for chicken soup with rice. You could have the allele for chicken soup with noodles. They're still chicken soup. That's why they're homologous. So I'm going to write down here where it says crossing over occurs, I'm going to say between homologous chromosomes. Because it does not just happen between any old chromosomes. So that's prophase 1. Very different than prophase in mitosis. Because of this crossing over event, we're exchanging genetic material. And you can actually see here that the red chromosome has a little bit of blue on it, and the blue chromosome has a little bit of red on it. So this is the product of crossing over. They trade little bits. In metaphase 1, this is still in meiosis 1, the microtubule assemblies are going to move everything around. If you remember in metaphase of mitosis, it was, we only had one X here, and then one X here, and then one X here. In other words, all of the chromosomes were down the metaphasic plate all alone. Here, they're not all alone. The homologous pairs are across from each other. So here's chromosome number 1 from mom and dad. Here's chromosome number 2 from mom and dad, homologous chromosomes. And then here's chromosome number 3 from mom and dad. So they're beside each other. They're 2 by 2 instead of single file. Everything else here is the same. They all line up on the metaphase plate, but 2 by 2 instead of single file. Then we have anaphase, the pulling apart. The pulling apart is exactly the same as in anaphase in mitosis. The only difference was in anaphase in mitosis, you wouldn't have entire X's. You wouldn't have entire copied, doubled up chromosomes being pulled. You would only have single chromatids being pulled apart. Notice as well, yeah, these chromosomes are not exactly the same anymore. The blue one has little tips that are red because it did crossing over, and the red chromosome has a little blue on the tips because it's done crossing over. Where did the blue come from? It came from here. Where did this red come from? It came from here. So they just did a swap, a trade of genetic material. And then we have telophase and cytokinesis. In telophase 1, we don't remake the nuclear membrane. So we don't make a membrane around here because we're going to do work with this cell like right away. So there's no need to put a nuclear membrane around the nucleus or around the DNA of the cell. So these cells are still 2n equals 6. So we still have 2 copies of a big chromosome, 2 copies of a medium chromosome, 2 copies of a small chromosome. So now this cell is going to do a cleavage furrow, so now it's going to be 2 cells, and each of these cells is going to undergo one more round of division. And so you see here a little bit of difference. We didn't do another S phase. If you look at the end of telophase 1, and then you look at the beginning of prophase 2, there's the exact same amount of DNA. Yes, it might be a little bit scrambled from what we started with, but it is the same as what we had in telophase 1. Pardon me. So we grow new spindles in prophase. We don't have to dissolve our membrane. Metaphase 2, we line everything up. Look at this. These guys are single file now. This looks a lot like metaphase in mitosis. The only difference being these chromosomes are not all exactly the same color. Why are they different? Well, look. Here's a red one with a tiny little blue nugget. Here's a blue one with a tiny little red nugget. Here's a blue one with a tiny little red nugget. Here's a red one with a tiny little blue nugget. So this guy, this is chromosome number 2. This is the homologous chromosome over here. Here's chromosome number 1. Here's chromosome number 1 in the other cell. And here's chromosome number 3 and chromosome number 3 in the other cell. So we have 2 of these cells, and they're both undergoing this process at the same time. So once the spindle fibers have attached and we've lined everything up on the metaphase plate just like before, we reel everything in. So we are pulling our chromosomes, or sorry, rather our chromatids apart. So they've separated from each other. And now we're going to reestablish the nuclear envelope, and the cytoplasm and the cell are going to split. What is that going to make for us? Well, it's going to give us 4 cells. But they are not 2n equals 6 anymore. These guys only have 3 chromatids in them. If you look at the start, we had 1, 2, 3, 4, 5, 6. We had 6 chromatids, so 3 pairs of chromosomes. Here we have 3 singlet chromosomes. So these are not 2n equals 6. These new cells are n equals 3. So these chromosomes are these cells are n equals 3, and they are all genetically different from each other. You look at they both have the top 2 have red chromosome number 2. It's fully red. They both have mostly red and a little bit blue chromosome number 3, the tiny chromosome. If you look at the big one, chromosome number 1, one of them has fully blue, but one of them has mostly blue with red tips. That means that these 2 cells are not genetically the same. Same deal down here. We have full blue for chromosome number 2, so that chromosome is identical. We've got, for chromosome 3, mostly blue with a little bit of red. That means those guys are identical. But then, for chromosome number 1, one cell has fully red, the other one has mostly red with blue tips. So that means these are unique chromosome, or unique cells. They are genetically not the same. And why is this n equals 3? Why do they have to be haploid? Quite simply because of the concept of fertilization. So if I have a sperm cell, we're going to go back to human beings. Here's our little sperm cell with n equals 23 chromosomes, and no, I'm for sure not going to draw them. And then we have an ovum. This is not to scale. An ovum is way bigger than a sperm cell for very, very important reasons. So it is also n equals 23. These are haploid. When they get together, they're going to form a zygote, and the zygote has the combined genetic info of both gametes. So then this cell is now diploid. Why? Because it's 2n. It means it has 2 copies of each chromosome, one from dad and one from mom. So this is the reason that all of the cells that are made from meiosis, all of the gametes, the reproductive cells, have to be haploid cells, because 2 haploid cells come together to make a diploid cell. So one last thing that we need to talk about in meiosis, something called nondisjunction. Now, it sounds like a complicated word. Disjunction just means to separate. So nondisjunction means a failure to separate. And this failure to separate can mess up the numbers of chromosomes that are in gametes. And this can happen in a sperm cell, or it can happen in an egg cell as they're developing. Certain egg cells generally are subject to this problem more than others, especially when a woman is older. I'll show you an example of this. Down syndrome is one of those problems that is caused by having the wrong number of chromosomes. And so that happens usually, it's the maternal age that is the big determinant. So that means the older a woman is when she gets pregnant, the higher the risk of getting having a child who has Down syndrome. So here is, let's see, this would be a 2n equals 4 organism. Oh, sorry, this is a 2n equals 2 organism, my mistake. No, it is a 2n equals 4. Sorry about that, guys. Because we're going to end up with n equals 2, or we should be ending up with n equals 2 sex cells, or gametes. So we've got a big chromosome blue, we got a little chromosome blue, so that's chromosome 1 and chromosome number 2. So we have 1, 2, 3, 4, so it's 2n equals 4. So in meiosis 1, remember, everything is doubled up. We align everything on the metaphase plate. Sometimes the spindle fibers have a little oopsie. And if the spindle fibers have a little oopsie, look what's happening. The big chromosome, chromosome number 1, both homologous chromosomes, so big blue and big red, are both being pulled to one side. And so when this meiosis 1 finishes, one of the cells in meiosis 2 is going to have extra copies. It's actually going to have all of the copies of chromosome number 1. And the other cell will have none of the copies of chromosome number 1. So if this cell, if this is an egg cell or a sperm cell, if it hooks up and combines with a normal companion cell, so I'm going to make, we should have a long chromosome and a short one. So this is a normal cell. It should be n equals 2, or yeah, n equals 2. We're going to have an offspring that has 3 copies of the big chromosome and 2 copies of the little chromosome. 3 copies of a chromosome means that this cell, this zygote, this fertilized egg, has too many copies of one of the chromosomes. When you have too many copies of a chromosome, generally that will cause the death of that zygote. It won't even make a baby. It'll basically self-destruct around the 4 or 8 cell stage. So that's probably a goner. The only situations where cells and babies can survive are chromosome or trisomy 21. I believe it's 13 and 18. Those are the only ones where it makes like a survivable cell. If you have trisomy of chromosome number 1, that doesn't even make a baby. That will just self-destruct because the genes that are on there, the gene products, the cell can't maintain proper development when there's too much of when there's one extra gene or one extra chromosome. By the same logic, if we get one of these other cells, this is missing chromosome number 1 entirely. It doesn't have a big chromosome at all. So if we have this situation, we're going to have 1 copy of the big chromosome, 2 copies of the little chromosome. Having only 1 copy of a chromosome is called monosomy. In other words, only 1 copy, 1 chromosome copy. We're supposed to have 2 copies of every chromosome, 1 from mom and 1 from dad. But monosomy means you have not enough of one of the chromosomes. You only have 1 copy of one of them. And so monosomy is also almost always fatal, meaning that the organism, the embryo, it won't even make an embryo. It'll call it quits. It'll self-destruct after a couple of cell divisions. So the 4 or 8 or 16 cell stage. So these are non-viable. And the only survivable ones, like in large number, is sex chromosome monosomy. So in other words, you can have this, where one of your parents didn't give you a sex chromosome. So maybe your dad gave an X and your mom had a problem like nondisjunction and made an egg cell with no X chromosome. So if you got an X from dad and nothing from mom, you have monosomy X. And that is survivable. If you have the opposite problem, if you have 2 copies of Y, like if there's a nondisjunction in meiosis 1 and you have 2 copies of Y, 2 copies of Y, if you have an X from mom and 2 copies of Y from dad, that's survivable as well. Generally, people who have non-balanced numbers of sex chromosomes, they can live normal lives. Sometimes there are minor disabilities that come with it. The major problem is sterility meaning that people who have unbalanced numbers like monosomy X, they will not be able to have children. They can't get pregnant, or they can't, well, these would be physiologically female, because the gene that causes maleness, quote unquote, is on the Y chromosome. It's called SRY, sex reversal Y. What if you have nondisjunction in meiosis 2? A better situation here in meiosis 1, 2 of the sperm cells or egg cells are going to form trisomy, and 2 of them are going to form monosomy. So this is, having a mess up earlier on is much worse. Here, this will, if it hooks up with a normal companion, will form trisomy of one of the chromosomes, and this will form monosomy. But the nice thing is, these ones are genetically normal. So that means that they will not have any problems. Is it possible that you could have both parents have a mistake? Yes, but it is exceedingly rare. So you could have a child where, let's say that mom had an extra copy of chromosome 1 and dad was missing, the sperm cell was missing chromosome 1. That would be okay, because you're still getting 2 copies of chromosome 1. You're just getting them both from your mom. Now, the chances of this happening are incredibly, incredibly rare. I'm not actually sure if that's ever been recorded, but it is technically possible where, again, one parent doesn't give you any copies of a particular chromosome, and your other parent gives you both copies. There would have to be non-disjunction in both parents. And they'd have to be non-disjunction concerning the exact same chromosome. So that's very rare. So I'm not sure, and you wouldn't notice. There would be nothing to go by. There's no clinical diagnoses. You would be a normal person. The only time that you'd be able to tell is if you got a DNA test like 23andMe, and they'd say, hey, wait a minute, both of your chromosome number 9, they're both from your mom. Your dad didn't give you a chromosome number 9 at all. Again, the only way that could happen is if mom had non-disjunction, either in meiosis 1 or 2, and gave an extra copy, and your dad would have to have non-disjunction somewhere and give you no copies of chromosome number 9. So you'd be a perfectly normal human being. You would have no clue. Little bit of an interesting kind of thing, theoretical thing to think about. So there's 2 ways that diversity can be given during the formation of sex cells. The first is recombination. Crossing over, recombination. And the second is, there is no reason why the red one had to go up and the blue one had to go down. These guys connect to the sister chromatids at random. What that means for us is 2 things. First, recombination drives genetic diversity. In other words, the fact that crossing over happens means that all of the 4 gametes that are formed are genetically unique. And the other one is called is that chromosomes go to the gametes randomly. That means that there is random assortment. The cell completely doesn't care. As long as there's one copy of each chromosome at the end of telophase 2, it's cool. So as long as in that original example, as long as we have n equals 3, one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, it literally doesn't matter. As long as you've got one of each, you're golden. In a human, as long as you have one copy of 1 through 22 and then one sex chromosome, you're golden. It doesn't matter if it's mostly blue and a little bit red, or mostly red and a little bit blue, or fully blue or fully red. It doesn't matter. That's the beauty of independent assortment, is that these chromosomes go in random directions. We just make sure each cell at the end of the day has one copy of each. So I hope that clarifies some things for you.